In chapter 13, we continue our discussion of the central dogma, where we talk about the process of translation, which is where we go from nucleic acids to amino acids, and then ultimately that amino acid chain is turned into a functional protein within a cell. This is a short chapter. There should only be 2 videos here. We are not going to talk too much about translational control or post-translational regulation. You don't need to know details there, except for a few sort of general things that I talk about. We're not going to do those too much, which makes this chapter even shorter. As part of this chapter, I wanted to go back and review some things from chapter 2, because chapter 2, we learned about the central dogma, and we learned about sort of some of the general ideas that we talked in more detail last chapter in chapter 12 with transcription, and then again in chapter 13 with translation. So I wanted to do that here since this lecture is a little bit shorter. And it's an important review, I think, because some of these concepts were a long time ago. I should have probably reviewed the defining characteristics of a gene in chapter 12, but quite honestly, I forgot that I wanted to do that. So just as a quick review, a gene must be replicated before it can be transmitted to the next generation. So this means that the genetic information in an organism has to be copied in order for it to be passed on. And as part of that replication process, there's a potential for variation. So changes happen during DNA replication that lead to variation within an organism. And we're not going to talk about this too much in this chapter, probably not at all, honestly, but we are focusing on expression. So a gene has to be expressed to produce a phenotype or some component of a phenotype. So recall that the idea of a phenotype, or the concept of a phenotype, is sort of the outward expression of a gene. Now, this could be a protein that you never see, quote unquote, see, because it's part of a metabolic process that occurs within a cell. Or it might be something that you do actually see. Maybe it is a gene that codes for pigment in eye color or pigment in hair color. But a lot of our genes are working as part of phenotypes that kind of all work together. And so you can look at this 2 ways. All phenotypes are coded by a genotype. And then the opposite way, of course, is that gene is expressed to produce a phenotype. Now, at the molecular level, most of the time, I would basically say all of the time when you're talking about a phenotype, you're going to be producing a functional protein. I just spelled that wrong. There we go, protein. We can also, though, produce what we call functional RNA molecules, and these are molecules that have a job within the cell, and the RNA molecule itself is sort of the phenotype because it is doing some sort of activity within the cell. Genes have a specific structure. We talked about that structure in chapter 12. Remember, it's a modular structure with the regulatory region, the promoter region, the coding region, and then of course that coding region is used to produce RNA. So if we're talking about the process of gene expression, this is often messenger RNA, although it can be some of these other functional RNAs as well. mRNA is what is directly used to make a protein. So the mRNA is important in the process of producing a specific phenotype. So just a quick review of our central dogma. We start with our DNA. We transcribe that. Remember, we're using not vocabulary words. We are using vocabulary words. We are using sort of language words here. We are going from DNA to RNA, which is essentially the same language. So when you rewrite something, you are transcribing it. You are writing it down. So we are just basically copying our DNA into RNA with some slight modifications. Remember that RNA has a slightly different sugar. It has a hydroxyl group on the sugar ring that DNA does not have. And then, of course, an RNA molecule contains uracil instead of thymine. And then the next step in the central dogma is translation. Remember, we're going from the language of nucleic acids to the language of amino acids. And one of the important things in the process of translation is that we need an adapter or a translator molecule in order to go from RNA to amino acids. And we'll talk about what that is in a few minutes. Recall that one strand of DNA is the template for the RNA sequence, and that is complementary to our coding sequence. And when that template is used to make a messenger RNA, we essentially get a messenger RNA that is the same as the coding sequence, except that it contains uracil. So the template is going to be complementary both to the coding and the coding messenger RNA. And then if you remember from chapter 3, and we'll do this again in a few minutes, each set of 3 bases in your messenger RNA codes for a specific amino acid. This is called our genetic code, or also called the triplet code. And it is that code in the mRNA that is used to produce that amino acid chain. Amino acid chains are also called polypeptide, poly meaning many. Peptide refers to the type of bond that links the amino acids. So these molecules are linked by a peptide bond, which is why we call that chain a polypeptide. And then to produce a phenotype, that polypeptide is going to be folded and modified into a functional protein. So originally, researchers thought that each gene encoded only messenger RNA. In actuality, genes can also code for other RNA molecules. In fact, most of the RNA that is produced in a cell is not actually messenger RNA, but it is functional RNA, another type of RNA, and these are referred to as functional RNAs. So these functional RNAs include some things that you're probably already familiar with, such as transfer RNA or ribosomal RNA, but it can also be regulatory molecules, such as microRNA or something called long coding RNA. We're not going to talk too much about long coding RNA. We'll talk a little bit about microRNA, and then we're going to come back to tRNA and rRNA. So just keep in mind that a gene may code for messenger RNA, in which case it produces a functional protein, but it may also code for these functional RNA molecules. Also, as a part of a review from chapter 2, I wanted to go through the one gene, one polypeptide hypothesis. So decades ago, when researchers were studying DNA as the molecular coding molecule, they put forth the hypothesis that each gene in the genome codes for one messenger RNA that codes for one polypeptide that produces one specific step in a phenotype. So essentially, if you have one gene, you're going to get one single polypeptide or one single protein that has a job within the organism. But more recently, as there's been more work done in the field of molecular biology, it has been discovered that, in fact, a single gene can actually make multiple but related polypeptides. And this goes along with something called alternative splicing that we'll talk about in just a little bit. But also it relates to those functional RNAs. Many genes do not code for polypeptides at all. In fact, it'll potentially code for those RNA molecules that have their own specific function. Only 5 to 10% of the human genome actually codes for messenger RNA. A lot of the genome actually codes for these other functional RNAs. So we're not going to say too much more about those RNAs until we get to the actual process of transcription. But just keep in mind that although we are focusing on this process of transcription in such a way that we learn all about those messenger RNAs and all about those proteins, the genome is actually coding for a lot of other things besides just proteins. So a quick review of process of transcription. We have our coding strand. It is transcribed by RNA polymerase into a messenger RNA. Now, transcription and translation in bacteria are all occurring in roughly the same location. Remember, in bacteria, there's no nucleus. And as soon as transcription begins and the messenger RNA begins coming out of the RNA polymerase, the ribosomes, which are the sort of worker proteins that produce the polypeptides, can actually attach almost immediately and begin translating that messenger RNA into a polypeptide. One of the major differences between one of the major differences in translation between bacteria and eukaryotes is that transcription and translation in eukaryotes occur at different times and in different locations. Eukaryotes have a nucleus. That's what is drawn here. And the nucleus obviously houses the DNA, and the DNA stays in the nucleus. So transcription occurs in the nucleus, and the messenger RNA is transported out of that nucleus. It is processed a little bit, and then a short time later, a ribosome will bind that messenger RNA and begin translation. So they are separated in time and space, although essentially, as soon as the mRNA leaves the nucleus, translation begins. So they happen very close together in time, but they do happen in different locations. Other than that, the overall process of translation is very, very similar in bacteria and eukaryotes. There are some slight differences in ribosomal structure in the sense that the proteins in the RNA are slightly different, but overall the ribosomes still function in essentially the exact same way. Now, this is really important, and we'll talk about this in a few minutes because it relates to the universal genetic code, the idea that all organisms use the same code and the same process for gene expression. We talked a lot about regulation of gene expression in chapter 12, and in fact, most of the regulation of gene expression occurs during the process of transcription. So it is energetically expensive for a cell to copy a gene into messenger RNA and then to produce a protein. So it makes sense that most of the control would happen before that gene is even produced. But there is a small amount of regulation that happens before translation. And so we might have a messenger RNA that is produced, and for some reason the cell does not want it turned into a protein. It turns out that there's a small region at the front of the messenger RNA, right before the start codon, called the 5' untranslated region. So this 5' untranslated region, also called the 5' UTR, is a binding point for regulatory molecules that can block the translation of this molecule. So if regulatory molecules bind this messenger RNA at the 5' UTR, there's going to be no translation. Now this might be a temporary thing or it might be permanent. If it's permanent, then the messenger RNA is going to be degraded and not turned into a protein. Our book doesn't go into a lot of detail on this, so I am not either, but I wanted to bring this in since we did talk about regulation so much in chapter 12. Another source of regulation is the binding of microRNAs. So as their name suggests, microRNAs are short RNA molecules, and these microRNAs can also bind to points in that messenger RNA that are going to block the ribosomes from binding to that messenger RNA, and therefore translation cannot take place. This is something called RNA interference. And again, our book doesn't go into a lot of detail here, so I am not either. Just keep in mind that you can actually regulate translation as well. So let's talk about the process of translation. Let's start with the components that we need for this process to occur. So the first thing we need is that messenger RNA that was produced in transcription. This is the code for our genes. We also need a transfer RNA. A few minutes ago, I said that we needed a sort of adapter molecule or a translator molecule that is going to actually interpret that messenger RNA and translate it into the language of amino acids. So the transfer RNA takes on this hairpin structure. It is a single-stranded RNA, as most RNA are. It's a single-stranded RNA, and it has a very specific shape that comes about due to complementary base pairing within itself. So it essentially folds back along itself so that it produces this transfer RNA shape. Now, one end of the transfer RNA has an anticodon that is complementary to the messenger RNA. The other end holds an amino acid. There is a specific enzyme called aminoacyl tRNA synthetase that actually locates the proper transfer RNA and attaches an amino acid to it. Lastly, we need the ribosome. There are two parts to the ribosome. We have the large subunit and the small subunit. And the ribosome acts as sort of a workbench on which the amino acid chain is built. The messenger RNA is threaded through a tunnel in the ribosome in between the large and small subunit. The transfer RNA is going to bring in an amino acid that is complementary to the messenger RNA site. Within the ribosome, there are some essentially pockets or grooves in the structure that have specific roles. The first site, the A site, the aminoacyl site, I like to remember this one as the amino acid site, just because it's easier to remember, but it is called the aminoacyl site. The amino acid site is where the tRNA actually comes in and brings the next amino acid. The peptidal site, or I like to remember it as the peptide site, is where we are actually going to grow our amino acid chain, and we'll talk about how that happens in a minute. And then the exit site, or the E site, is where the transfer RNA that no longer contains an amino acid actually leaves from the system. So again, the ribosome is the ribosome is made up of two parts, but the ribosomal subunits are made up of both RNAs, ribosomal RNA, that's one of our functional RNA molecules, and proteins. So the subunits are sort of these massive structures that are made up of these two components. The larger subunit is called the 50S subunit in bacteria, and the 60S subunit in the eukaryotic system. So this just means essentially that the eukaryotic ribosomal subunit is slightly larger. That S refers to sedimentation. So one of the ways that you can characterize something on size is by spinning it very, very quickly and looking at where it falls within the centrifugation tube. So that's all that the S means. It is made up of, both of them are made up of specific ribosomal RNAs as well. You can see that eukaryotes have slightly larger RNA molecules. And then if we look at the small subunit, it is in fact smaller. It's made up of smaller proteins, or less proteins, and smaller ribosomal RNA. The ribosome is one of the most highly conserved structures. This means that throughout evolutionary history, the ribosomal genes really haven't changed. Now, if you think about why this might be, if a ribosome is required for gene expression and you have no functional ribosome because it's accumulated a mutation, you don't have an organism. It cannot live and grow. And so ribosomal RNA and those genes for the ribosomal proteins really haven't changed much. The major differences come between the big domains, between bacteria, eukaryotes, and archaea. But even those are still pretty similar to one another. So down here, this computer image on the bottom, we've got the large subunit, the small subunit, and then we've got our pockets, the A, the P, and the E sites. There you can see those anticodon. These are actually the tRNA in those A, P, and E sites. And then here we have the anticodon at the bottom of those transfer RNA molecules. Here's a few different ways that transfer RNA are depicted. We have these different types of models. They're just different levels of structure and simplification, because things get a little messy when you start drawing in ribosomes and messenger RNA. I like this image on the left because it shows all of that complementary base pair stranding within the single-stranded molecule. If you follow the ribbon around, you'll see that it is in fact a single strand that is folded back upon itself. Down here, we've got the anticodon. Each transfer RNA is going to code for one amino acid. So this anticodon is specific for an amino acid, and it is complementary to the messenger RNA. So our messenger RNA has a code. The complementary anticodon is found on the transfer RNA. And again, those amino acyl synthetases recognize the anticodon and it knows which amino acid to bring to the transfer RNA. Our book, for the remainder of this section, really uses this really simplified transfer RNA molecule that shows the anticodon on one end and the amino acid on the other. And that's just to keep things nice and clean in their images. Remember that Watson and Crick discovered this, along with a few others, discovered the structure of DNA. And very shortly after that, Crick proposed a method, basically this method, although the details were a bit fuzzy, for the process of gene expression. And he's the one that actually coined the phrase central dogma and proposed this adapter molecule that actually does the translating between the RNA sequence and the amino acid sequence. So this transfer RNA is also important because it helps set the reading frame of our messenger RNA. If you remember from chapter 2, we read our triplet code in sets of 3. This is a non-overlapping code. And so your transfer RNAs are going to, if they could, line up sequentially over that messenger RNA. They don't overlap, they move one after the other. Transfer RNA is a fantastic example of structure and function. So when I teach Biology 101, one of the big things that I talk about is structure and function. That the structure and function. One of the big themes in biology is this idea that the function of a system is uniquely and specifically related to its structure. And the other way, the specific structure of a system, in this case the transfer RNA, allows it to do its job. So without that anticodon in the amino acid, we don't have this translator molecule. So let's move on to how the cycle of translation actually works. Now, like many things that we've talked about, I believe that this is better seen in motion. I have linked in our required lecture videos for this chapter a video, an animation of translation. There's all over YouTube if you want to see this moving. And I think that you should watch this as a moving process because this is 3 parts, the transfer RNA, the messenger RNA, and the ribosome all interacting in a continuous motion to produce the amino acid chain. So let's walk through this cycle. So we are going to start with this is sort of in the middle of a process, but we start with an amino acid. Transfer RNA brings in the second one. So it'd come in from here. And then as part of the ribosome's job, it catalyzes the bond, the peptide bond between the amino acids. And then the system shifts and the empty transfer RNA is moved off. Another amino acid comes in. That bond is transferred to the incoming amino acid, and our amino acid chain continues to grow. So if you look at this, we can actually see the movement and the shift. So the amino acid comes in to the A site. We have the growing polypeptide at the P site. Now I know you can't see them. It's just our book did this to make it a little more simple. But you've got that big ribosome subunit behind, you've got your messenger RNA below, and then you've got your exit site. So in this process, that peptide chain is physically moved off of the tRNA and transferred to the next amino acid. And then the whole system shifts. So now this amino acid in tRNA that was in the A site is now in the P site. The A site is empty, and that empty transfer RNA is in the E site. Now another amino acid comes into the A site. The amino acid chain in the P site is transferred over, and that transfer RNA that is empty leaves out the E site. It can be recharged by aminoacyl synthetase and an amino acid added back. Then, of course, we have our shift. We have this amino acid chain at the P site. A site is empty. E site now has an empty transfer RNA. So if you look at the messenger RNA, you can see that this whole system is shifting along the messenger RNA. And then, of course, we've got the last one. This amino acid comes in to the A site. The chain from the P site is going to be transferred, and then we just continue until we get to a stop codon at the end. There are three major steps in translation. First is initiation or start of translation. This is signaled by the start codon. This encodes the amino acid methionine. In bacteria, this is called F-met, and it is brought in by an F-met tRNA. It is a specially modified methionine because methionine can also occur within the code as well. The only difference between the addition of the methionine and the addition of the other amino acids is that the AUG methionine, the start codon methionine, is actually brought to the P site. And this is because when the next amino acid comes in to the A site, that methionine has to be transferred over. So the AUG codon with the methionine is brought in, the AUG code signals the bringing in of the methionine. And so translation is initiated when you get the messenger RNA, the small subunit, and the methionine all in place. Then the large subunit comes in, and after that, we can begin producing our amino acid chain. So in the cycle of elongation, we really sort of already talked about this. We have our amino acid chain building in the P site. A new tRNA comes in with a new amino acid. That chain is transferred to the A site, and then the whole system shifts just a little bit. And then your tRNA exits and a new tRNA comes in. And this cycle, again, just continues. Eventually, we hit a stop codon. So stop codons are unique in that there is no transfer RNA that recognizes a stop codon. So we get to the end and there is nothing new to bring in, and so our polypeptide chain is terminated. There are some proteins that come in at this point called termination factor proteins that sort of disassemble the whole system, release that messenger RNA, release the polypeptide, and that polypeptide chain is then going to be modified and processed into a functional protein.